Good morning and good afternoon. Thank you all for joining the When Accuracy Matters SMART Sequencing in Clinical Research and Diagnostics session of the Global PacBio Summit. My name is Lori Aro, and I am the Senior Director of Clinical Genomics here at PacBio. The agenda for today is I will take you through a couple of slides to set the context of HIFI sequencing and how we generate our highly accurate sequence data, and then share with you the several targeted sequencing solutions that with some examples from work that we have done internally as well as work that has been done by some of our collaborators. And then discuss briefly the workflows and multiplexing solutions that PacBio offers for targeted sequencing. And finally, we'll end with a Q&A session. So to start, I think it's important to set the context, even though probably most people are very familiar at this point with our CCS sequencing mode and our HIFI read generation. But I thought it would be helpful to discuss how we get such accurate single molecule reads. This comes with our circular consensus sequence method. With our SMART-Bell template, each insert is sequenced multiple times, creating subreads, which are aligned to generate a single molecule HIFI read as shown on the right. The sequencing errors here indicated as red dots are random and therefore are washed out by the increasing number of passes around the SMART-Bell. The consensus accuracy for a HIFI read with just 10 passes is 99.9%. HIFI reads then enable complete coverage of the regions of interest, allowing us to easily map reads, detect variants, and phase haplotypes, as shown on the right side of the slide. This is in contrast to short read technology that has sequencing gaps which make it hard to detect and map variants, as shown on the left. Our ability to have base level accuracy allows you to differentiate between real variant and errors. For example, with only 22-fold HIFI coverage reads, gives you an accuracy score of Q51, and you see 99.5% of the reference sequence genes, considerably better than when compared to other long read technologies, and even better than when you combine other long read technologies with short read polishing. So the advantages of HIFI smart sequencing are that we can sequence amplicons or inserts as large as 20 kb and in some cases even longer. We like to say that if you can amplify it, we can sequence it. And the consensus sequencing you get are very high accuracy, QV50 or higher. PacBio sequencing has minimal GC bias, and with no DNA amplification. HIFI reads from a single molecule that have a high single molecule accuracy of greater than 99%. All of the advantages of HIFI sequencing allow you to capture your region of interest in high resolution. We have complete uniform coverage for your targets, even in low complexity regions such as repeat expansions, promoters, and flanking regions of transposable elements. We are able to characterize the full spectrum of genetic variation, including SNPs, structural variation, indels, alternative transcripts, low frequency variants, and haplotypes. HIFI sequencing is a cost-effective, comprehensive way for accurate variant detection. Now I'd like to share with you some examples of how our customers are using HIFI sequencing on clinically relevant targets. Let's start with targeted sequencing. As I mentioned previously, smart sequencing can generate very long amplicons, up to 20 kb in length. These long amplicons allow for greater primer design flexibility. It's not necessary to design primers within the target region. The benefit of amplicon sequencing with HIFI reads over short read sequencing and Sanger sequencing is that the reads are longer and therefore can be completely sequenced in one read, whereas other methods require primer walking across the region with assembly after the sequencing. Those shorter reads that we just mentioned, amplicons must be assembled to make a complete read of the target area of interest, but with HIFI long reads, there is no assembly required, greatly simplifying the workflow and reducing the risk of dropouts and improving your accuracy. PacBio supports 3 targeted sequencing approaches. The first is PCR-based for amplicons, where you can quickly and high numbers of samples on a single SMRT cell, also showing the ability to phase across alleles. Second is capture-based with PCR, where you can build large panels of targeted genes of interest, and large meaning up to megabase sizes. And finally, an amplification-free approach that we call our no-amp approach, allowing you to amplify hard-to-sequence regions in your genome that are difficult with traditional PCR-based approaches. I'm going to highlight both long-range PCR and no-amp approaches, really demonstrating PacBio's flexible options that you have the ability to pick and choose what works in your laboratory. Our ability to generate these long, accurate reads of 20 kb or more in any sequence context allows for successful sequencing of difficult regions. The genes I'll focus on today are shown here and include some with pseudogenes, such as SMN1, GBA, and CYP21A2, and genes with repeat expansions, such as HTT and FMR1. We target with either long-range PCR or no-amp target enrichment methods. The target regions can range from as small as less than 1 kb to as large as 16 kb. So I'll start with SMN1 and SMN2, associated with spinal muscular atrophy and a debilitative neurodegenerative disorder. These genes are located in a highly complex region of chromosome 5, shown in the upper right, with large 500 kb inverted duplication. SMN1 and SMN2 are nearly identical, and most assays focus on a single variant between the two genes in exon 7 that determines expression level. Together with our collaborator, David at Dor Yesharim in Israel, we have developed a PCR-based assay for a 16 kb region in and around exon 7 in both SMN1 and SMN2. The surrounding region includes enough differences to accurately map the read to either gene. Then, focusing on exon 7, gene conversions can be identified, as shown in the lower right. This work was recently described in an e-poster at the ASHG meeting that occurred last month and is available at the PacBio.com website. So here is an example of a sample with an SMN2 to SMN1 gene conversion. This sample had 1 allele mapping to SMN1 and 2 alleles mapping to SMN2. One of those 2 SMN2 alleles had the exon 7 functional SNP, meaning a conversion from SMN2 to SMN1, as shown in the bottom of the IGV map. This type of gene conversion looks like an SMN1 in counting assays and is actually fairly common, as shown on the right, and can indicate the 2-0 genotype, or a silent carrier. Here, a sample with another SMN2 to SMN1 gene conversion, but this is of a larger region, so it maps to SMN1, making 3 SMN1 alleles. It's linked to a variant called the Luo variant, easily identified in the top panel, along with other variants for each allele. In the bottom left is shown the results of a recent test sample with a high number of variants that were all correctly identified, including 3 samples with ambiguous qPCR results due to a SNP under the qPCR primer, which was discovered in this sequencing data. Now, moving to the second gene, HTT, associated with Huntington's disease. The disease severity is related to the number of CAG repeats. We targeted this gene with 2 methods, long-range PCR, targeting a 5 kb region, and our no-AMP CRISPR-Cas9 method, targeting a similar 4 kb region. The no-AMP protocol details for targeting the HTT repeat region are available on the PacBio.com website. The goal of the study was to determine the number of repeats per allele and interruption sequences, and to identify surrounding variants and phase with repeat alleles. We looked at 3 Coriell samples with alleles having moderate to high expansions. As shown here, the PCR and no-AMP methods had similar number of repeats for the normal and expanded alleles, with over 100 repeats for the sample, matching the available information on Coriell genotypes. In comparing the methods, PCR generates many more reads per sample as expected, whereas the balance between normal and expanded alleles is more even with no AMP, and there would be no PCR issues associated with that method. Here on the left, you can see reads from both methods map as expected, showing phasing of repeats and other surrounding variants. On the right are 2 ways of showing the number of reads versus the number of repeats. On the top, we have histograms for easy visualization of alleles, and below, that is what we call our waterfall plot, with bars for the number of repeats color-coded by repeat motif. Both assays show CAG repeats followed by a smaller number of other motifs. Now I'll show you just a peek at our early development work on GBA associated with Gaucher's disease, and the complexity here is that it is a very similar pseudogene, GPAP1, located just upstream, leading to frequent recombination. We're looking at targeting the entire 10 kb gene and pseudogene and a bit more of the surrounding area to determine phasing of variants and detect deletions. At the lower right, you can see mapping results from the control sample. We're taking a similar approach with CYP21A2 associated with congenital adrenal hyperplasia, which is located within a region having 4 duplicated genes in tandem, as shown at the left. This leads to a high level of recombination in this area, which makes it very difficult to characterize with short reads. In order to detect and map these recombinant events, here we have targeted the entire 3 kb gene and pseudogene, plus a sizable amount of the surrounding region, as shown on the right. Finally, I'd like to show a few slides on our recent collaboration with ASURAGEN. ASURAGEN is a company with a proven expertise and technology in amplifying through difficult sequence contexts, spanning a wide range of GC content, as shown on the left with their AmplideX PCR technology. So when we combined the ASURAGEN AmplideX PCR technology with PacBio's HIFI sequencing, we chose to look at FMR1 first. So sequencing of the very difficult CGG repeat in FMR1 showing the number of reads versus the number of repeats for 3 samples with expanded alleles going up as high as 300 repeats. Now looking at the waterfall plots for FMR1, on the top of each we can see 2 AGG interruptions for the normal alleles for all 3 samples, indicated by the orange vertical lines in the first sample's expanded allele as well, whereas there is no interruption in the expanded allele for the other 2 samples. So now that I've had a chance to highlight both the long-range PCR capabilities as well as our no-AMP capabilities and our emerging capabilities with the ASURAGEN AmpliDex technology, I wanted to spend a moment talking about multiplexing and the workflow for our 2 approaches. PacBio offers multiple multiplexing and barcoding solutions for AMPLICONS, allowing a large number of libraries to be pooled and sequenced simultaneously on a single run on the system. Our multiplexing is supported with 4 barcoding options. We have our barcoded M13 primers, the barcoded universal primers, barcoded adapters, and barcoded primers. Here is an example workflow using pooled barcoded amplicons. This example shows specific targeted primers with barcodes incorporated directly into the primers, but barcodes can be incorporated into the amplicon via a 2-step tailed primer approach, as in when we use the M13 approach. PacBio has a set of 384 barcodes that are optimized for smart sequencing. And these barcode sequences can be added to the ends of the PCR primers, generating potentially hundreds of barcoded amplicons, which can then be pooled into one SMRT library, prep, and sequenced on one SMRT cell. Data is then demultiplexed by barcode and analysis is performed separately on each sample. So PacBio's Amplicon workflow is quite simple. It's a 3.5-day workflow with 2.5 of those days are off instrument and only 1 day on instrument, allowing for high level of instrument utilization via serial runs. The no-AMP targeted sequencing method is also quite easy to implement as well, and it's basically a 5-step process with step 1, extracting DNA, step 2, block 5' and 3' ends, step 3 is the CRISPR-Cas9 digestion, and then 4 is ligation of the barcoded adapters, and then finally the step 5 is running on the SQL system, and then analysis is conducted on the SMRT link using HIFI reads and visualization of results. So what can you do with one 8M SMRT cell? For targeted sequencing with amplicons, you can run upwards of 1,000 samples, ranging in size from 1 to 10 kb. And when you're running that level of multiplexing, your sample price becomes quite modest, so it's a $1 to $2 per sample. And for no-AMP sequencing, for enriching those hard to amplify targets, you can multiplex up to 48 samples on one 8M SMRT cell, and getting down to a price between $82 to $118 per sample, making targeted sequencing quite efficient and affordable on PacBio. So with PacBio, it's easy to go from DNA to target to answer quickly and efficiently. You have from library prep, your complete template prep can be done in 6 hours. We accept a variety of sample types and insert sizes, the size selection options to enrich for longest inserts, and we have flexible multiplexing and barcoding solutions. For smart sequencing, we can sequence very rapidly from a half an hour up to 20 hours. You can serially process multiple SMRT cells. and you have walk-away time of up to 4 days. Data analysis is completed using a variety of analysis methods available through Smart Analysis and PacBio's DevNet community, as well as open source software and advanced data visualization and mining tools. So the benefits of sequencing for AMPLICONs are a simplified workflow, a single primer set per target up to 20 kb with no assembly required. Smart sequencing for AMPLICONs and targeted sequencing is very cost effective with high throughput for sample multiplexing, multiple and flexible barcoding options, flexible AMPLICON size pooling, and reduced chance for dropout and no assembly required. The reduced time to results, which is very important, is flexible run times and hands-off time. And of course, our HIFI accuracy and resolution, capturing all genetic variation, including low frequency variants, generating those HIFI reads of greater than Q50. I'm going to conclude with just highlighting a couple of additional presentations that are going to be happening over the course of the next few days that you might find interesting, as they are also oriented towards clinical research. The first is third-generation sequencing for diagnosis of rare disease by Dr. Neil Miller. Second is long-range PCR meets long-read sequencing tandem technologies for tackling the toughest genes in carrier screening by Gary Latham from ASURAGEN, highlighting some of the collaborative work that we have done. Third is increasing solve rates for rare and Mendelian disease with long-read sequencing by Dr. Jenny Ekholm, who will be highlighting some of our whole genome work as well as our NoAMP. And then finally, the comprehensive analysis of thalassemia alleles, a universal approach to thalassemia carrier testing by Dr. David Cram of Berry Genomics. And he will be highlighting some of the development work that they are doing on the Sequel system for their clinical applications. And with that, I would like to conclude the recorded remarks and open the session up for questions and answers. Thank you.
Hello, everybody, and welcome. I have Lori Aro, who's our Senior Director of Clinical Genomics here at PacBio. My name is Nick Cisneros. I'm the Senior Director of the Field Application Scientist team here in the Americas. And if you can and have any questions, please go ahead and enter those now in the chat option, and we'll go ahead and get those answered. So it looks like we have a question right here off the bat. What is the best kit to PCR greater than 10 kb products, and how to reduce PCR bias?
That's a really good question. And actually, I think it is dependent upon the target that you're looking at. And so if there is a specific target that you would like us to get back to you with specifics around, we'd be happy to do that. So I don't have the kit information in front of me. So Nick, maybe you can forward that question directly to me and I will respond directly to the questionnaire, and we can find out exactly the target they're interested in, get them a recommendation based on our utilization.
Okay, perfect. Yeah, we can do that. We'll definitely follow up on that regard. The next question is, what is the amplicon size range for the long-range PCR approach?
Boy, we have customers who are actively doing a 16 kb amplicon and multiplexing that on the Sequel 2 system at over 384 samples for a multiplexing level. We have other customers who are actually multiplexing high levels of amplicons upwards of 20 kb in some of the pharmacogenomic genes. So on long-range PCR, we have definitely demonstrated greater than a 20 kb amplicon works very well.
Okay, and we have time for about one more question here. So if your questions haven't been answered, Don't worry, we'll follow up with you as we get those coming in. So please continue to put your questions in, and we'll follow up with you. For the SMN1 and SMN2, how many samples can be multiplexed on a single SMRT cell?
Yeah, so the assay that we've been working on for the SMN1 and 2 genes is around a 16 kb amplicon in size, and that's the one where we've demonstrated that on a Sequel II system you can multiplex up to 384 samples on a single SMRT cell.
Okay, perfect.
And really driving the cost per sample down to a very cost efficient, and certainly more cost efficient than the traditional method of, say, MLPA.
All right, fantastic. Thank you, Lori, and thank you all for attending this session. That's all the time we have for questions today, and we will follow up with any questions that didn't get answered. Thank you guys for your time. Thank you, Lori, for your presentation. That was great, and we'll see you all soon.
Thank you.